Hi everyone, I hope you are all doing well. So if you can see from the title, we have a bit of a bigger lecture here. We are rounding out our series on plant anatomy by looking at these final plant structures. So before we get into this, we're going to do a quick review of our root and stem development, and then we'll jump back in. So let us start by looking at our roots. And I'll keep a little diagram down here, right? Here's our root. And if you remember, we said that all of our root tissue, at least our initial root tissue, derives from the apical meristem. That is the tissue. Meristematic tissue is that it's made up of actively dividing cells. It produces other tissues. All right, so our root tissue, our roots, all of those tissues are going to be produced by this apical meristem. And it produces four main tissues that later differentiate into other tissues. So it starts out with our apical meristem produces root cap. Now that's not going to differentiate any further. That's just what it is. And if you recall, our root cap is at the tip of our roots, right here. And then we'll shade it in. And this root cap is going to protect the growing root. Right, so this root is growing down through soil. As it does that, these cells get damaged. But they're protecting all the other cells further up in the root. So this apical meristem continually produces new root cap cells to replace those that are lost. And right back here is where we find our apical meristem. So that's the first. The second is our apical meristem produces what we call protoderm. And this protoderm is going to develop into the epidermis, which is the outer layer of cells of our root. And in our roots, this epidermis, some of them also form root hairs, which are the primary way through which water and other nutrients enter our roots. Right, so here the outer layer of our cells is the outer layer of our root is the epidermis. And sometimes we have that epidermis forms these extensions that are root hairs. Right, and that is where most of the water enters our root. Okay, then our apical meristem, I don't need the arrow, produces ground meristem. And this ground meristem, these cells are going to mature into what we call cortex. And there's a special part of the cortex called the endodermis. So we have our epidermis on the outside here, and then moving inward, this region right here would be our cortex. And that cortex is mainly those parenchyma cells, the all-around cells. They're going to act as storage here. They're storing different nutrients. And the inner part of our cortex, that right there is our endodermis. And our endodermis has that important role. It has, why are you doing that? It has the Casparian strip, which is that waterproof strip. So what this is doing is interior, in the inner part of our cell is where our vascular tissue is. That is the xylem and the phloem tissue, and it's the xylem that's going to transport water and minerals up throughout the rest of the plant. Well, this Casparian strip of the endodermis makes it that water just can't diffuse across into the xylem and start traveling upward. it has to go through a cell if it wants to get into the xylem. So this is the way that our plants can regulate the amount of water that they take in, and minerals as well. Right, so it's important, and it's all due to this endodermis and this Casparian strip. And it's an important mechanism that allows our plants some measure of control over the water and the minerals that they're taking in. And then finally, we have our apical meristem produces procambium, and our procambium produces pericycle, primary xylem, primary phloem. If we are a dicot, vascular cambium. If we are a monocot, pith. So this is dicot, monocot. So, unfortunately, I have it drawn very closely to our diagram over here. Sorry about that. So we said here's like the outer part of our root, and this interior part is what we call the stele. And if we look at the stele, top down, right, so this section right here is our stele. They say the outside of our stele is the pericycle. And our pericycle is responsible for the production of branch roots. So we have our main root coming down. We're going to have a secondary root branch off from this main root. That new secondary root is going to form at this pericycle. It just starts kind of branching off and growing and growing and growing. So we have our pericycle. We then, this is going to be a dicot root. Then we will have, since it's a dicot, in the interior of our dicot is our vascular tissue. So this is our xylem, and our xylem is transporting water. And then around the xylem, we have phloem, and that is the vascular tissue that is transporting photosynthate, or sugar, primarily. And then, in between them, right here, is our vascular cambium. And that is going to produce secondary xylem and secondary phloem. Right, so that's producing our secondary xylem as this root is going to thicken through what we call secondary growth. That is done through the production of this secondary xylem and secondary phloem. So this vascular cambium produces more xylem this way, more phloem this way, and our root gets thicker. So that's our dicot root. Our monocot root has essentially all the same structures except for the vascular cambium. So our monocot. But the arrangement is different, right? So we have our pericycle right here. But instead of this vascular tissue in the center, our vascular tissue kind of exists as these separate, not really bundles, but just separate tissue. So we have xylem, phloem, xylem. Forms a ring around our root. And the interior here is pith. And pith is similar to our cortex tissue. It's going to be mainly those parenchyma cells that are acting as storage cells. Right, so that is our monocot root cross-section. Okay, I think that's good. Hopefully that all makes sense to you guys. We're now going to take a look at our stems. And our stems are very similar. right? It all starts, just like our roots, with the apical meristem. What is different is that we're not producing in our stems the root cap, right? So we don't have to worry about that. But what we do produce, just like with our roots, are the protoderm, and that's going to form the epidermis. And in our stems, that epidermis is covered with a waxy material called cutin, which is going to waterproof the stem and waterproof our plant. We then have the ground meristem, which is going to produce our cortex. Now, in our stems, our cortex is a little different. In the roots, it's primarily those parenchyma cells. In our cortex, our stem is all three of those main tissue types, right? We have I think it's parenchyma. I think it's an E. For parenchyma, parenchyma, and parenchyma. So we have all three of our tissue types in our cortex, and this is our storage. This sclerenchyma is going to be the really structural support. Right, it's just cell walls, the cells are dead. And this is also structural support, but it's a more flexible support. The sclerenchyma, these cell walls are with lignin. The cell walls are thickened with cellulose. Makes a difference in how rigid they are. And then we also may find in our cortex some secretory cells, which are going to produce resinous compounds that can act for defense. Right, and that's our ground meristem. Oh, and our cortex, we're also going to form from this in our woody plants a cork cambium. And that is going to produce new cork tissue, which is the outer layer of our bark. So I'll show that in a cross section as well. And then lastly, we have, just like with our roots, our procambium, and that is going to produce our xylem tissue, primary xylem, phloem, vascular cambium, if we have our dicots, and our pith tissue, which is produced in both. Right? So very, very similar. And if we're looking at a cross section, we can do in here, this will be our woody dicot down here. Our cross section. In the center here is going to be our pith. And then essentially, just like a tree, you'll have those rings. And that is xylem tissue. And we'll say right here at the edge of this ring is our vascular cambium. Initially, there would have just been primary xylem here and primary phloem up here. But as this tree is growing and thickening, this vascular cambium is producing new xylem or secondary xylem. And out here is, kind of looks kind of wavy like that, is new phloem tissue. There will be a strip that goes all the way around here. That is the cortex. At the edge of the cortex would be the cork cambium. And then you have your cork or bark or outer layer on the outside here. And the bark includes everything from our epidermis all the way to our phloem tissue. That's our woody dicot. And then if we're looking at our dicot, looks a little bit different. Again, you got your epidermis, you have your cortex, you don't have your core cambium, and then you would have in the interior here your pith. And then the big thing with our herbaceous dicots is that it has these vascular bundles around here. And our vascular bundles running through them is our vascular cambium. And then on the interior here is our xylem. Exterior here, oops, we'll go down here, is our phloem. Right? And then our monocots are the easiest to draw because they're just your same thing, epidermis, and then pith tissue in the center, and then just random xylem and phloem throughout. And if you recall, our monocot stems, they don't grow through that secondary growth. They don't get thicker through the production of new xylem and new phloem tissue like our dicots. But they can get thicker through the replication of parenchyma cells. And that is diffuse secondary growth. And that occurs in some of our monocots, particularly our tree-like monocots like our palms. Right? They get a bigger stem, and that's occurring through the replication and growth of parenchyma cells. Okay, so that is an overview of the development of both roots and stems and some of their anatomy. I want you guys to know this. I know it's a lot of different vocabulary, but once you get it down, it's very similar between the two types. There's only a couple little differences that you need to know. All right, back to the lecture. Okay, so we're going to start by looking at leaves. Our plant leaves are, you can think of them as photosynthesis factories. Right? That's their primary function of the leaf is photosynthesis. And again, photosynthesis is a process by which a plant takes in carbon dioxide and water and using energy from the sun combines them to create glucose or a sugar molecule. And it's that sugar molecule that is then used in this process called cellular respiration. It gets broken down, and from that it provides energy to the cells. It's the basis of metabolism for all organisms. So photosynthesis is very, very important, and the leaves are the structure that is responsible for photosynthesis. They also, a secondary function is cellular respiration, which I just talked about. And with some of our leaves, they get modified for other functions. And I have two examples there. We have black locust leaves get modified to become what we call thorns, and they aid in protecting the plant. Some of our vining plants have modified leaves called tendrils that help support the plant as they vine up other structures or other plants. But when we're talking about leaves, we're primarily talking about photosynthesis. Now, if we look at our leaf anatomy, we see there's a couple layers to our leaf. Just like with our stems, the outer layer is the epidermis, right? And with our epidermis, we have a cuticle, which is this thick, waxy surface that's covering it. That's going to reduce water loss. Below that, we have mesophyll cells, which make up the bulk of our leaf. Now, we have two different types of mesophyll cells. We have palisades mesophyll, which are primarily the main site of photosynthesis that you see right here. Then we also have spongy mesophyll, and in this layer is where we see most of our gas exchange. Right? So gases, carbon dioxide and oxygen come in to the leaf, and we'll talk about how they get in there in a second. And this gas exchange and oxygen leaves the leaf, and this occurs in this spongy mesophyll primarily. We also have veins, leaf veins, and these are made up of vascular tissue, that's xylem and phloem tissue, and those leaf veins run throughout the leaf. A quick little aside, it's the development and proliferation of veins within leaves that scientists believe is why our angiosperms, our flowering plants, were able to outcompete the gymnosperms, the pines and conifers, right? Because if you have more veins in a leaf, you get more water to your leaf, you can then have more photosynthesis, create more food, etc. Quick little aside. But anyway, getting back to our leaf, we have very, very important structures that we call stoma, if we're talking about one, or stomata. And they are openings or pores found primarily on the underside of leaves, in most of our leaves. And now these stomas, or stomata, I should say, have two guard cells that surround each one, and they can regulate its opening and its closing. Now this is very important because it's through the stomata that the plant is able to take in carbon dioxide. It's also with the stomata that we have what is referred to as transpiration, and that is the loss of water in the leaf. Now, transpiration can be a good thing. It can help regulate leaf temperature, and it also is responsible for actually pulling water up through the plant, and we'll learn about that a little bit later. A downside of this process is that when plants start to lack water, where there's not sufficient water, they will close their stomata to prevent further water loss. But when they do that, they also prevent further photosynthesis because no longer is carbon dioxide coming into the leaf. And that can have greater ramifications leading to cell death, plant death, etc. So very, very important, the stomata. Now, if we're looking at classifying more leaf parts, some of the terms that you'll see is the leaf blade. That's the main body of the leaf that you see here. There is something called the petiole, which is the leaf stalk. Right? It is this essentially a stem that is going to connect the blade of the leaf to the proper stem. Not all leaves have petioles, as we'll see in a little bit. There are stipules, which are little leaf-like appendages found at the base of a petiole. Our leaves have a midrib, which is this main vein that runs up through the middle of the leaf. This midrib is vascular tissue, it's xylem and phloem, but it also contains some structural tissue to provide some support to the leaf. And like I said, we have veins that branch off from the midrib that you can see there. Another part of our leaf is the leaf margin. That is the outer edge of our leaf. And you will see that different leaves have different types of margins, right? And we have different examples here with dentate, which are toothed, serrated, which are variations of the teeth that point upward, lobate, like an oak leaf, etc. Part of your lab is going to be to kind of look for these. So these margins, you don't need to know all of these. I have them up here as just reference. This is something, if you guys get into botany, you will need to learn some of these terms because this is some of the ways in which we identify different types of plants by looking at leaf margins. So again, you don't need to know all these specific terms. No leaf margin is the edge of the leaf, but I'm not asking you to know the specific margins. You should know these differences, though, how a leaf attaches to the stem. We have petiolate, which is our leaf has a petiole. Again, that's the stalk of our leaf. So in a petiolate leaf attachment, we have a petiole that attaches the leaf to the stem. If we have a sessile leaf, there is no petiole, but the leaf itself is attached directly to the stem. Another important distinction of our leaves is that we have simple versus compound leaves. With our simple leaves, there is just one leaf, or there's one body to the leaf. With our compound leaves, our leaves are composed of smaller leaflets, right? So in a simple leaf, this is just a single leaf. Each of these is a single leaf as well, but you see these leaves are broken up into smaller leaflets. Now, a way to tell a simple from a compound leaf, right? How to tell that, or really to tell a simple leaf from a leaflet, is that all leaves, they have a bud, or it'll be a bud scar at the base of the petiole, right? So if you don't see this bud here, like you don't see it here, this is a leaflet rather than a leaf. And we have different arrangements of our compound leaves. You can have trifoliate, pinnate, where you have your, this actually is referred to as a rachis in our compound leaf, and you see leaflets branching off from each side, or palmate, where your leaflets all originate from this origin point right here. Again, I don't expect you right now to know these differences, but you should know the difference between a simple leaf and a compound leaf. And we're also going to look at leaf arrangement. Again, this is important if you get further into botany and you're looking at plant identification. Our leaves can be arranged on the stem in different ways. The two most common are alternate and opposite. In an alternate leaf arrangement, you have one leaf on either side of the stem as it works its way up. With an opposite arrangement, you will have leaves on either side of the stem at the same location, right? So you can see with alternate, we have one leaf branches out here, then we move up a different leaf. With opposite, you see two leaves that are opposite of each other on the stem. We also can see, this is less common, whorled leaves, where there's multiple leaves that branch out from that same point. And lastly, our leaves can have different types of venation. So we can have parallel venation, where we have our midrib and our other veins that run parallel to that midrib, kind of starting from this same point here near the petiole and working their way up. This is more common in our monocots. And more common in our dicots is this net venation, where you have your midrib that runs up and then lateral veins that branch off from that midrib. Okay, so those are veins. I am going to pause this because I need to take my dog out for a walk. All right, so moving on, we have buds. Buds are an undeveloped shoot or flower. Now they are composed primarily of meristematic tissue. Again, that meristematic tissue is made up of the actively dividing cells. It creates new tissue. And our buds are found in axils of leaves, or at the terminal end of shoots. So an axil is this angle that you see here between the leaf and the stem, and you'll find it here at the base of that leaf, which we refer to as a node, or you will find it at the tips of shoots. Now our buds, we have three types: vegetative buds that are going to develop into shoots, flower buds that are going to develop into flowers, or mixed buds that can develop into either one. And our buds can be active or dormant. Many plants will produce buds that then quickly become dormant, and they will remain dormant until certain environmental conditions or physiological conditions in the plant. I should be doing this as a slideshow. Okay, so that's it. That's all you need to know for buds. We're now going to look at flowers. Now, our flowers are the reproductive part of angiosperms. And remember, they are really what make angiosperms, right? Angiosperms are flowering plants. That's the big differentiation that we make between them and gymnosperms. Now, flowers are really just modified leaves and stems, and they're very important because they facilitate sexual reproduction. I'll go in at a later lecture when we look more in depth in pollination. I'll talk a little bit more about sexual reproduction and its importance to, from an evolutionary standpoint, why sexual reproduction is so important and why so many organisms practice it. But for now, you should just remember that that flowers are the structure, the mechanism that facilitates sexual reproduction in plants. And importantly, after fertilization, portions of the flower then develop into the fruit, which will bear the seed or seeds of the plant. Now, our flowers have a number of different parts. The receptacle is the thickened portion of the stem from which the flower will grow. Receptacles will be larger in some species than others. They may play a bigger role, as we'll see later on with the strawberry. We have sepals, which are modified leaves that encircle other parts of the flower, and collectively the sepals will be called the calyx. We then have petals, which again are modified leaves that you find above the sepal. Collectively, all the petals are called the corolla. And our petals are often brightly colored to attract various pollinators. They may contain nectaries that are going to produce a sugary substance to attract pollinators. And then the sepals and the petals collectively are called the perianth. Now, we also have stamens, which are the male part of the flower, and the pistil, or carpel, which is the female part of the flower. Now, these are the stamens right here, and the stamens consist of a filament, which is this long stalk, and the anther, which sits atop the filament, and it's the anther that is going to produce the pollen. And it's the pollen that contains a sperm cell, or eventually two sperm cells, that combines or attaches to the carpel over here, and we'll talk a little bit about pollination in a second. But that pollen is essentially contains the male gametes, or the male sexual reproductive cells. Now, the female part of the flower is the pistil, like I said, or the carpel. And it has three parts: a stigma, style, and ovary. The stigma is this receptive surface that receives the pollen. A style is the tube that connects the stigma to the ovary. And then the ovary contains undeveloped ovules, which will eventually develop into seeds. And within each ovule is going to be an egg cell that gets fertilized by the sperm cells in the pollen. So, talk about pollen. These are examples you see up here. This is kind of zoomed in on an anther. And pollen consists of a vegetative cell and one or sometimes two reproductive cells. And it's also protected by this outer layer that can take different shapes that you see. Now, the vegetative cell is eventually going to grow a long tube, and we can refer to it as a tube cell. It forms this pollen tube. And the reproductive cells are going to divide into two sperm cells. Whereas the ovules, right here, are in the ovaries, and they are going to give rise. Here's a better example. Here are ovules in the ovary, and they are going to give rise to seeds. They are actually going to be what the seed is. And within each ovule, sometimes you can have just one ovule in an ovary, sometimes you have multiple. Within each one, you will have a number of different types of cells. Importantly, you will have an egg cell, and you will also have these two polar nuclei that are eventually going to give rise to the endosperm, which is that food storage of the cell. Now, our pistil, our carpel right here, it can be made of a single carpel, which consists of your stigma, style, and ovary, or it can be made up of multiple ones that get kind of fused together. And we refer to that as a compound pistil. Right? So you see here, this would be a compound pistil where you have multiple carpels that fuse together at the bottom. And there are a number of plants that have compound pistils. One example is the cucumber that we'll look at a little bit later. And you can actually see each of these individual carpels and how it gets fused. We can also have a different position of the ovary in flowers. It could be what we refer to as superior, so it's above the receptacle, half inferior, where it's halfway in the receptacle, or inferior, where it's kind of hidden, buried in the receptacle. Okay, so those are the structures of the flower and a little bit about how some of those structures get positioned. The actual process of pollination and fertilization is, at its basics, fairly easy. Our pollen grain is going to land on a stigma through various, one way or another, wind pollination through a pollinator insect. That pollen grain is going to grow a pollen tube, right? The male reproductive cells need to get into an ovule where the egg is. The only way to do that is to go through a long tube. So this pollen grain grows a long tube, which we call the pollen tube. Two sperm, the reproductive cell in our pollen, divides into two sperm cells. They make their way down the pollen tube into an ovule. One sperm cell goes and fuses with the egg, and that's going to turn into the embryo of the plant. And the other sperm cell fuses with these polar nuclei, and that is going to develop into the endosperm, the food store for our eventual seed. This entire ovule becomes a seed, and like we said, within an ovary there could just be one ovule, one big seed, multiple ovules, each developing into a separate seed. Now, a couple more things about our flowers. Our flowers, we can classify them in different ways. So we have what are referred to as complete flowers, which contain all four parts of the flower: sepals, petals, stamens, and pistil. Or we can have incomplete flowers that lack one or more of these parts, right? So in flower B, it's lacking the stamens. It doesn't have the male reproductive part of the flower. Within our incomplete flowers, we can also have other classifications. So we have what are referred to as perfect flowers. And our perfect flowers have both stamens and pistils. Now they may be incomplete, they may be lacking sepals or petals, or they may be complete and have all of them. But importantly, our perfect flowers have both stamens and pistils, both the male and female reproductive part of the flower. If we don't have perfect flowers, we can have what are referred to as staminate flowers, where they just have stamens but no pistils. You can think of them as the male flowers. Or we can have female flowers, which are pistillate flowers. They have pistils but no stamens. Now, if we have those types of flowers in a plant, we can have monoecious plants, which have both staminate and pistillate flowers in one individual plant. Examples are alder, corn, walnut trees. Or we can have dioecious plants, where we have one individual plant will have all pistillate flowers, all female flowers. Another plant will have all male flowers. So you have different male and female plants. And there's different examples of those: pistachios, asparagus, holly tree, that we'll take a look at. Right? So again, our monoecious plants have both male and female flowers on the same plant. An example is the corn. The tassels of our corn consist of the staminate flowers, the male flowers. They are producing pollen. The silks that form in our corn, these are all essentially carpels or pistils that are sticking out from the corn, right? Each one of these the long silks is a style. At the end of that style is a stigma, that surface that's going to receive the pollen. As the pollen falls from the staminate flowers, either from the same plant or other plants, it's going to land on each individual stigma. And we're going to grow a long pollen tube. Sperm is going to come down and fertilize the ovules, or the eggs in the ovules in each one of these ovaries. And that's how we get our corn. Now, that's a monoecious, right? We have both the male and female flowers in one plant. We also have dioecious plants, where we have separate plants that are male and female. Examples are holly trees. We have trees that just produce female flowers and trees that just produce male flowers. So our flowers can take on different arrangements. Right now, they can be born singly on a stalk, and that would be a solitary flower. Think of like a sunflower. or they can be arranged in clusters that we call inflorescences, or a flower inflorescence. There are many different types of flower arrangements and inflorescence that you can see. Okay, all right. Buddy, could you not make so much noise? I'm not asking you to know all of these, but I do want you to know that what an inflorescence is. Now, as part of the inflorescence, you have what is called the peduncle, which would be the stalk of the inflorescence. And you also have the pedicels, which are the stalks of the individual flowers. Again, I'm probably not going to test you on that specifically, but it's something to know, again, going further on in your academic career. Here are just more examples of inflorescences and different types. Just to show you as reference, you're not going to need to know the different types. A couple more examples. Okay, so now we're moving on to the seed. And remember that I said the seed is a mature ovule, right? Those ovules are found in the ovary, which is a part of the pistil of our plant. And the seed is composed of three main parts. And we've already kind of learned this stuff already, right? We have the embryo, which is the mature plantlet that is formed from the union of the male and the female gamete. And gamete is just another word for the sex cells. All right, so it's the fusion of the sperm and the egg, forms a gamete, and that then divides and eventually is going to form this embryo. And that embryo, the main parts that you've learned about are the radicle, the plumule, and the cotyledons, and that plumule can be the hypocotyl and the epicotyl. We also have storage tissue, the second big part, and that storage tissue could be endosperm. It could be storage tissue that is part of the cotyledon. We have seeds that are called albuminous seeds that have a large store of endosperm. Those are typically our monocots, right? Think of that corn seed. We also have X-albuminous seeds that have no or only a thin layer of endosperm. That's more of our dicot type seeds, right? Have big cotyledons, only a little bit of endosperm. And then we have the seed coat, also called the testa. Now this is formed from the integuments, which is just the outer layer of the ovule, and their seed coat is going to protect the embryo from damage. Now lastly, we're going to talk about fruit. Our fruit is the mature ovary plus associated parts. Again, think of that, the female part of the flower. Think of that pistil. You have stigma, style, and then the ovary. And within the ovary are those ovules, and those ovules are going to be seeds. And the ovary is going to turn into what we think of as the fruit. Now, the fruit protects the seed and also helps disseminate it, right, through various means, being ingested by different animals, etc. Now, our fruit is not just fleshy. We tend to think of fruits as, like I have there, grapes, berries, apples, peaches, pears, etc., that are more fleshy. But fruits also include nuts, legumes, grains. Those are all technically fruits because they are the result or formed from ovaries, flower ovaries. Now, the pericarp is an important part of our fruit. That is the ovary wall, and it's the pericarp, the ovary wall, that is going to turn into or kind of form whatever type of fruit that we have, and we'll see that in a little bit. But again, just as your reference that I have up here, here's our pistil, stigma, style. This is the ovary. This is what's going to turn into the fruit, and it's this ovary wall that is going to turn into different types of fruit depending on the species, and we refer to this as the pericarp. Now, we have two big designations of fruits: simple fruits and aggregate fruits. We're going to start talking about our simple fruits. And our simple fruits have a single ovary formed from one flower. So our simple fruits are formed from one ovary. Now that ovary may be a compound, that pistil may be a compound one, so it has multiple ovaries that are fused, but it's a single main ovary. And now our simple fruits can have multiple ovules or seeds within them. You don't just need one seed. We have three common categories of simple fruits: fleshy, dry fleshy, and dry. And this is where, like I said, our pericarp plays a big role, because it's how that pericarp develops, what it develops into, that is going to determine whether it's a fleshy fruit, a dry fleshy fruit, or a dry fruit. So with our fleshy fruits, our pericarp develops into succulent tissue. And examples are berries, blueberries, grapes, peppers, tomatoes even. Hesperidiums, which are your oranges, lemons, limes, your citrus fruits. Or peepos, which are cucumbers, melons, squashes. And here's our cucumber, and here you could see in the outline here that this is a fused compound pistil here. So each one of these little sections is an individual, keep going, would have been an ovary from a single carpel that gets fused into a compound pistil. So it's kind of neat because you can kind of see those individual sections. And within them would be all the different ovules and then develop into all the different seeds. So those are fleshy fruits. We then have dry fleshy fruits, where in this case our pericarp, some parts remain succulent, other parts get dry. And the two big examples are our drupes, or our stone fruits, such as our peaches, plums, and cherries. And our pomes are apples and pears. We then have our dry fruits, and these are fruits in which the entire pericarp is dry at maturity. And within our dry fruits, we have we can break it down into two further categories. Our dehiscent fruits, which split or dehisce at maturity. So think of different legumes, peas, and beans that split. And your indehiscent fruits, which do not split at maturity, and we'll see them in a second. But within our dehiscent fruits, we have legumes, like I said, different types of, again, you don't need to get into the details here, but different types, and they're all some variations on what actually, how we classify them. But I want you to know the main classifications: fleshy, dry, and then within dry, dehiscent, and indehiscent. And like I said, indehiscence, the fruit does not split at maturity. And examples are achenes, which is the strawberry, grains, nuts, acorns, chestnuts. Samaras are the maple fruits that you see there. The interesting thing about achenes in a strawberry is that the actual strawberry fruit is that. What you would think of as, some people think of seeds. That's the fruit. This whole big fleshy thing that we're eating, that we consider the strawberry, is actually the receptacle. It's the modified stalk on which the flower sits. Because our strawberries are an example of an aggregate fruit. So we had our simple fruits that's made from one ovary, and then we also have aggregate fruits, which are made from several ovaries. So if you look at the strawberry flower, you see that there are individual pistils or individual carpels situated in a single flower on this kind of fleshy receptacle. So each one of these ovaries, or each one of these pistils, gets pollinated, and that ovary is going to develop into the fruit, which is the tiny things that you see there. And it just happens to grow on this large receptacle that is fleshy and juicy and delicious. So examples of our aggregate fruits are our strawberry that I talked about there, blackberries, pineapple, raspberry, mulberries. Those are all examples of aggregate fruits. So they're made from several ovaries, whereas our simple fruits are developed from one single ovary. And that is it. So hopefully that's not too overwhelming. I know it's a lot of different terms, but if you take each one and each step, you guys will have this down. That is it for this week. Next week we're going to have a review, and then it's your first test. So hope you guys are doing all right, and I will talk to you next week.